Hi everyone, this is Yan. I'm a PhD student at the University of Missouri. I would like to present my poster. The title of my poster is A Comprehensive Suite of Tools for Allele Discovery and Phenotype Improvement in SoyKB and KBCommons Frameworks for Crops. There are 5 tools in my poster, which are the Variant Calling Pipeline, the Allele Catalog Pipeline, the Allele Catalog Tool, the Genomic Variation Explorer, and the Multiple Allele Discovery Tool. First, we will start with the Variant Calling Pipeline. The Variant Calling Pipeline is developed using the SnakeMake workflow engine by incorporating the Burrows-Wheeler Aligner and Genome Analysis Toolkit. The Burrows-Wheeler Aligner can align raw sequencing reads in FASTQ files and pass the aligned reads to Genome Analysis Toolkit for processing in parallel in order to obtain SNPs and indel in VCF files. This pipeline is available on GitHub and it can be used in both standalone server and high-performance computing cluster. Next, I would like to present the Allele Catalog Pipeline. The Allele Catalog Pipeline is developed using the same methodology as the Variant Calling Pipeline by integrating bigger imputation tool, SNP-EFF functional effect prediction tool, and data aggregation scripts into the SnakeMake bioinformatics workflow engine. The Allele Catalog Pipeline takes in metadata file, VCF file, and GFF file to perform imputation and functional effect prediction on SNPs and indels, then utilize data aggregation scripts to generate Allele Catalog datasets. The Allele Catalog datasets are the data that power the Allele Catalog tool. The Allele Catalog tool has a data query page which consists of 2 search windows. The Search by Gene IDs windows allows users to input multiple genes with each gene per line to perform search, while the Search by Accessions and Gene ID windows allows users to insert multiple accessions and 1 gene to perform query. After users click the search button, they will be redirected to a result page which has the Allele Catalog data of the queried genes. The allele information is grouped based on unique alleles, functional effects, and amino acid changes. The accession counts are summarized based on unique alleles groupings and improvement status. If users are interested in a particular allele distribution, they can click the accession counts to visualize the exact accessions that has the allele distribution along with metadata. The variant position in the Allele Catalog table is also clickable to connect with phenotypes in the Phenotype Viewer. The Phenotype Viewer consists of a phenotype selection accordion, 6 buttons, and a phenotype data table. Users can select phenotypic traits at the phenotype selection accordion and click View button to view data at the phenotype table. If users are interested in the phenotype distribution, they can click on the phenotypic trait heading in the phenotype table. The quantitative data will be presented as violin plots, while the qualitative data will be presented in the form of bar plots. Next, I would like to show the Genomic Variation Explorer. In the Genomic Variation Explorer, there are 2 components, which are the promoter region component and the copy number variation component. In the promoter region component, users can input multiple genes to perform query to obtain results, which include transcription factor binding sites, binding transcription factors, and gene binding sequence in the binding TF table. If users are interested in a transcription factor binding site, they can click on the binding TF to visualize the sequence logo figure of the gene binding sequence and a breakdown table of the gene binding sequence. SNPs and indels in the gene binding sequence can link to phenotypes with the Phenotype Viewer. In the copy number variation component, users can input multiple genes to perform query. The result tables include information of the queried genes in a table, the CMV regions and accession counts in a table, as well as neighboring genes of each CMV region in each distinct table. The buttons in the CMV regions and accession counts table can connect copy numbers with phenotypes in the Phenotype Viewer. The distribution of quantitative phenotype data is shown in violin plot, while the distribution of qualitative phenotype data is displayed in bar plots. The last tool that I would like to present is the Multiple Allele Discovery tool. The Multiple Allele Discovery tool main user interface allows users to select dataset, input genes of interest, and provide phenotype data to perform MADIS calculations. The phenotype data consists of two columns, which are the accession IDs and phenotypes encoded in 0 and 1 for wild-type and mutated phenotypes accordingly. In the two steps MADIS calculations, combinations of two variant positions are calculated in the first step. Based on users' selected variant positions in the first step, combinations of 2 to 7 variant positions are calculated in the second step to obtain best variant position combination that have the best score and explain percentages. The combination of variant positions in the table can be linked to the Allele catalog tool to visualize the accession and allele distributions and assist researchers in select accessions of interest. Clicking on the numbers of matching or mismatched wild-type and mutated phenotypes can generate a pop-up table for users to view the exact accessions, positions, alleles, and phenotype information. All the presented tools are publicly available and users can use the links listed here to access the tools. I would like to thank United Soybean Board for providing valuable funding to support our research. I also would like to thank members in USB Project Group for providing expert guidance and support throughout this research. Thank you.